Hello, everybody. This is Miss Peachy again from WCA Biology B. Here we are in Unit 8, Lesson 4, and this is your final portfolio for this school year. It is Animal Phylogenetic Tree Portfolio, and I have changed so many of the portfolios that they have offered through this curriculum, but I actually like this one. So we're going to keep it intact. It is divided into multiple parts. I'm going to kind of explain that to you here and what you're going to need to submit to me. I have actually created a document that I'm going to put on my Student Hub page that's going to give you an outline of everything you need to complete or turn in so you know what you're actually being assessed on. So at the end of this is going to make not as much sense at the moment, but it'll make sense later. You will submit 2 character matrices, 2 cladograms, and the responses to these questions that I have put below here that you can copy and paste into your document. And we're going to explain what the character matrices are and what the cladograms are in this lesson today. This one's actually going to take a little longer because we have to go through some examples. So if you look at the course outline, the portfolio is divided up into 3 lessons, lessons 4, 5, and 6, with the submission of the actual portfolio done in lesson 6. But what it's talking about first is constructing a cladogram. If you remember, a cladogram is a specific type of phylogenetic tree, kind of shows relationships of organisms. So you can kind of see what characteristics they have in common. And if you can look at enough characteristics, you should be able to construct kind of like a family tree with different organisms to see how closely related to each other they are. Thinking about a family tree. In your family tree, you start generally with, think about it. You're like, okay, you've got your mom and dad, you've got brothers and sisters, potentially, you've got your grandma and grandpa who have, on both sides, you have 2 sets of grandparents, and then you're going to have uncles and aunts and cousins on both sides. So it can get pretty complex pretty quickly. And you can see the degree of relatedness. So your parents are literally like 1 degree of relatedness away from you. Same with your siblings, right? But when you go beyond that, your grandparents are 2 degrees of relatedness away from you. There's 2 generations difference between them. Same thing with cousins and uncles and aunts and things like that. So we can kind of look at how closely related one of your family members is to you based on where they lie in the family tree. And we can look at how closely related organisms are to each other based on where they lie in the phylogenetic tree. So it's a similar type of process, but also looking at relatedness as well. Ultimately, what you're going to be doing is you're going to be looking at an alien species and you're going to be constructing a cladogram, which is a type of phylogenetic tree, from the characteristics you're observing of these particular fictitious alien species. So you'll construct a cladogram and you're going to kind of categorize or organize all the different ways in which the different characteristics that you can observe in your aliens, and that's kind of what we're building on, but we're going to get to that point in a little bit. So it talks first about family trees, which you and I just kind of talked about a bit ago. You got your grandparents. Grandparents had, let's say they had a son and a daughter, and each one of them got married, and then so this is one of their sons, this is their son and his wife, and they had 2 children, and this is their daughter and his wife, daughter, and her husband, and they had 3 children. And this is just a small part of a family tree, right? So you can look at characteristics that they have in common. Look at the kids have red hair and freckles that they inherited from their mother. So we can look at inherited traits and how they're passed on from one generation to the next. We can look at the blonde hair from their daughter here, which is passed on to both daughter and son, but the brown hair from dad is passed on to the daughter. So again, we're looking at a observable characteristic, which we call derived characteristic, and how that characteristic is passed on to a different generation. We can add more generations to this. We can go beyond it, add cousins and aunts and uncles and things like that to make this even bigger. And that gives us more information. Some words we're going to see in this section are morphological, phylogeny, convergent, evolution. So here we see a kangaroo and a cat. These guys are not super closely related, but they do have some characteristics in common. We can see what types of characteristics do they share. Well, the kangaroo and the cat both are furry, so they share the characteristics of having fur. They both have four limbs. They both feed their young milk and give birth to live young. So they've got maybe more characteristics in common than you might think of before you really sat down to make a list. Unfortunately, there's some things they don't have in common. For example, cats have really sharp teeth because they eat meat. There are carnivores. In fact, they are, what do they call them? Like they're obligate carnivores. They have to eat meat. Kangaroos, on the other hand, have flat teeth that allow them to eat grass. So those differing characteristics tell us that they are, somewhat different from each other as well. And it's these differences that suggest that cats and kangaroos broke off from a common ancestor because they don't share all characteristics in common, but they do share some. The study of phylogeny is the study of evolutionary lines of descent. We are looking at how things are related to each other. Scientists determine evolutionary relationships by looking for similarities in the fossil record. Can't see everything that ever existed right now, so you have to look at a historical record of things that existed in the past. And the fossil record provides us, a historical record. It's not a complete historical record by any means. It's very incomplete, but it does provide us with some information. One of the earliest methods scientists used to determine evolutionary relationships was by comparing morphological characteristics between living organisms and the fossils of extinct species. This is just what things look like and how they're built. Morphology is the study of form and function, like, do you have an opposable thumb? Do you have four limbs? Are you bipedal, or are you, quadrupedal? Two legs, you walk on two legs, you walk on four legs. So not just about form, but also function. So I can have four limbs, but I could be walking on two legs versus walking on four legs. That's a different in function, right? So it's about both how you're built, but also how you work as well. Species that are more closely related tend to have more characteristics in common. Of course, this approach does have some limitations, for instance, in which two species can have very similar characteristics, but not be that related. And remember that we also look at other lines of evidence like DNA that helps to kind of back up these observations so they can kind of see, you know, verifying it using DNA and stuff like that as well. Let's see here. So it does talk a little bit about convergent evolution as well, how just because two things look alike or have similar form and function doesn't necessarily mean they are related to one another. If organisms evolved in similar ecosystems and similar environments, they had the same kind of pressures put upon them, they had the same types of things they had to adapt to, so they could very likely be very similar to one another simply because of the environment in which they evolved. That doesn't mean they actually at any one time were related to one another, but they kind of parallelly evolved separately, but similarly because they had similar types of challenges, similar types of biological niches they had to fill, similar types of foods they had to eat and stuff like that, right? So how do they distinguish between similarities that arise from a recent and common ancestor and convergent evolution? And as I mentioned, there's that whole fun thing called DNA. You can look at the number of shared amino acids, the shared genome sequences and stuff that exist in DNA, which we talked about in a previous lesson. And if you came to a live lesson recently, you would have had a class on that because we just did a really cool activity in class about that. That actually provides, supplemental information. It helps to confirm or to not confirm whether or not two different organisms are really closely related to each other. I'm trying to move through this one in a rapid but also meaningful way because there's actually a lot of content here to cover. Okay, so when they use the evidence from DNA and from fossil records, they create what are known as phylogenetic trees to show evolutionary relationship. So we can see here, anytime there's what's called a node, I call it like an armpit. I don't know. It's called the node is the exact term, but for some reason it just seems like an armpit to me. So anytime they have like this area, this shows, or this is representative of a common ancestor. Now, one of the things I recently read about is when you do a cladogram, which is what we're going to be constructing, this is a hypothetical common ancestor that should be here. When we're constructing an actual phylogenetic tree, which varies a little bit from a cladogram, you have to actually have a common ancestor that you place at each of the nodes as well. So what we can see here is that species C and species D, they are related to each other. Their most recent common ancestor was here in this first node. They also were related to species A, but that was a lot longer ago. So if you want to think about this as here's brothers and sisters, and that's mom, and that's grandma, but they're more closely related to mom than they are grandma. Now that's kind of using a family tree as an analogy again, but that's the same kind of concept we're talking about here. And as we look at this, you go further back in time to the left. Phylogenetic trees start with a common ancestor that links all organisms in the tree. Many phylogenetic trees also use different line lengths to represent different lengths of time. That's something I talked about previously. So long line lengths represents long periods of time, and short line lengths represent shorter periods of time. So as you can see, the chimpanzee is related to tribe Panini much more recently than the gorillas were related to tribe Gorillini, right? So that's kind of where that line length becomes somewhat meaningful. Okay, so that was kind of, let's see here. Just moving on to the second part of this lesson, we have cladogram, derived character, and phylogeny. And it talks about a derived character. I have read it as a derived characteristic. It is a characteristic, so I don't know why they're calling it a derived character unless that's something that I made a mistake on. But essentially, it's a trait that separates an organism from its ancestors. It's something that an organism has that the ancestors don't have. And they're using these derived characters here: cells, backbone, legs, hair, and opposable thumbs. Do these organisms possess these specific traits? In this example, slugs possess cells. They do not have a backbone, legs, hair, or opposable thumbs. They're invertebrate animals. They don't have backbones. Human beings have all of them. Catfish have just cells of backbone, but no legs, hairs, or opposable thumbs. Frogs have cells, backbone, and legs, but no hair or opposable thumbs. And the tiger has all of them except for the opposable thumbs. So you can construct this on a line, kind of a line to show how closely related they are to each other. If you have all the characteristics that are shown, then you belong to the right side of the line, human being. If you have fewer characteristics, you kind of go down the line to where on the left side, that organism has the fewest of all of those derived characteristics, because slugs don't have any of these guys, they only have cells. So this is a very simple cladogram, and that's kind of what you're going to be constructing with this alien species as well. You're going to provide a diagram or a picture that looks a lot like this. You can either make a computer kind of picture like this if you want to do it like on Word or something, or you can draw it. That's fine too. It can be drawn as long as you give me a picture of your picture. That's fine too, but that's what you're going to be creating as well. That's part of it. In fact, the two big pieces you're going to be creating as far as your actual, like the actual things you're, the word is just escaping me, not things, but the actual like products that you're making here will be a table like this and a picture like this. And then you'll have questions to answer. So I hope it's seeming to be not quite so confusing right now. I just give you more practice with this, and then we are moving on to lesson 5, which is portfolio 2. So essentially, you're going to be getting some more practice with cladograms as you go through how to read the cladograms and how to make the cladograms. You're going to watch a video on them. Again, another little table here. This is going to show you these characteristics: jaw, lungs, fur, gizzards, and feathers. And those are your organism: lamprey, sea bass, antelope, bald eagle, and alligator. Right? And then you are given an example to create your own table and your own from this particular cladogram. So you're kind of working backwards here. More practice with cladograms. Those you can kind of go through on your own. And then we're going to talk about a different concept, analogies, character of polarity, and homologies. So let's look at analogies and homologies first. Characteristics that are similar because of common ancestry, but that instead evolved as adaptations to a similar environmental challenge. Versus homologies that are characters that are similar because they are inherited directly from a common ancestor. Let's see here. So analogies versus homologies. For example, if you look at all the four limbs of bats, cats, dogs, humans, whales, horses, and horses, the bones are very similar. Okay, all the organisms have a long bone at the top and two smaller bones in the middle, wrist bones and hand and finger bones. The bones have slightly different shapes in each organism, but the bones are there. These bone structures are homologous because these organisms are all mammals and have inherited the same bone structure. So this would be considered homologous. So I don't have a picture of that. They probably don't. But so homologous means that they share the characteristic because they've inherited that same trait. It's passed on from one generation to the next. Now, the flippers of a whale look very different from the forelimb of a cat, which looks very different from the hand and wrist of a human, but the bone structure inside is the same, and that's why it's considered homologous, because it's the bones themselves that are inherited, not necessarily the function, because whales use their flippers for swimming, right? And bats might use their wings for flying, and people use their arms for picking things up and writing. So the function isn't necessarily the same, but the structure is the same. Analogous means that the function is the same even when the structure is not the same, right? So when you look at the wings of a bat versus the wings of a bird versus the wings of an insect, they're very, very different structurally. They're not built the same at all. There's no similar bone structure, nothing like that, but they're all used for flying. So they have the same purpose, the same function. So part of this challenge here is you're going to determine if these traits are analogous or homologous. So do they have the same form but different functions, or do they all have the same function but not necessarily the same form? Different form or same form but maybe different functions is homologous, and same function but possibly not built the same is considered analogous. Okay, so that's important. And again, more examples of different types of cladograms. So now we move on to the actual thing you're going to be doing. And man, we've already gone into this quite a bit, but guys, I just want to make sure you have the background information you need. So you are going to create a cladogram of some aliens. This is a species of xenite, which is an imaginary animal that have been found on a planet in another solar system. Based on the order of the rock layers in which the fossils were found, species A appears to be the oldest and probably the most primitive. There's no evidence to support the relative age of the other species of xenite, so we don't know how old the rest of them are. We just know that A is likely the oldest, okay? So what you're going to do is you're going to look at these characteristics: spines, eye stalks, tail, and mouth. And you are going to be creating a table that probably will look very similar to, where is it? This table. Xenite A, B, C, D, get it? Those are your characteristics, and you're going to put yeses and nos in the box. Okay, that's the first thing you're going to do. And then, based on the table, you're now going to create a cladogram. We're going to go back to, is it the previous one? I showed you how to do that, creating the cladogram. There's a lot of pages in this, I apologize, but pretty sure. This is kind of what you're going to do. It's just going to be a line. We know our oldest one, right? They told us. So the oldest goes all the way to the, what's that side on the picture? But it's all the way to the left. I'm a mirror right now, so all the way to the left. And the youngest goes all the way to the right. Okay? And then you put the characteristics on the bottom. If it has that characteristic, it has to appear after it. So tigers have hair. That's why they appear to the right of where the hair is. Humans also have hair. They also have opposable thumbs, so they appear to the right. Is this making sense so far? So that's the first thing you're going to do. Create this table for your little alien guys, and then a cladogram. Then I'm gonna kind of give you a heads up. We go on to lesson 6, which is slow loading today. I don't know why. And they're saying, surprise! We found a new one. We have a species F here. Woohoo! Sorry, should have given that to you right away. So now you have to create a new table, include species F, and a new cladogram including species F. And I want you to do both of these. I don't want you to do just one with species F in it because I really want you to see how it changes if you have new evidence. That's important. So you're going to construct two tables. The first one excludes species F, and the first cladogram is excluding species F. Then put species F into the mix and see kind of how things change, create a new table and a new cladogram. They're not that hard, so you should be able to do those, okay? So once you've done that, then you're going to answer these questions, and please make sure you're complete with your answers, right? If it says why, or if it says how, or if it says explain, you need to do that. I've assigned points, making sure that you get points for doing that. So question 1, explain how you created your new cladogram and identify which derived characters were homologies or analogies. So what you could do is when you make your little table, you could just make a separate column that says homology or analogy. So then you have that information in your table, or you could provide a separate list here. It really doesn't matter. Explain the evolutionary history of the organism shown by your tree. Explain how you use new evidence to revise your cladogram. That's important. Okay. Question B is kind of hard. Explain the evolutionary history. So who came first? Who came second? Who's the oldest? That kind of stuff. Okay. Question 2, how did you choose to structure the table of derived characteristics of xenites? What were the rows and columns and what order did you choose? When you added xenite F, did you reorganize your data table? If so, why? Question 3, what features were the most difficult to place in the cladogram and why? Question 4, do you think your answer is the only right answer? Explain. Question 5, give an example of a piece of new evidence that would change your cladogram. What new information might cause you to change your mind on this? And that's it. I don't think this is super difficult to do. If you have questions, let me know. I'm always here to help if you need to meet in a live lesson room to kind of go through this more in person, but try it out on your own first, see what you come up with. Don't come to me empty-handed. If you have a question, at least try to see if you can construct a cladogram first and then kind of come to me and say, this didn't make sense. I would really appreciate that. But I'm always here to help if you need it. Good luck, everybody, and I will see you in the next recording.